Alright people, if you can't tell that this is an eye, then I cannot help you. Alright, so what do we have to look at? Fibrous layer, sclera and cornea. Cornea, 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 sclera, 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 vascular layer. Choroid has our blood vessels and melanin to give it the nice black. Choroid means black, and black helps to catch photons of light so they don't scatter. Yeah, that's the inner layer. So here's the vascular layer here. Here, you can see it back here. So the vascular layer has this choroid in the back that has the blood vessels and helps to trap light. And then in the front it has these ciliary muscles that are attached to the ciliary zonule, which is also known as suspensory ligaments. So the whole thing together is the ciliary body, and the ciliary muscles are going to hang on to these ciliary zonules, which are grabbing onto the lens and either flattening it or bulging it. Okay, so that was vascular layer. Choroid layer, ciliary muscle, ciliary zonules, iris. Iris, oh, oh, iris, oh, oh, iris. Okay, so see how the pupil's a hole? That's the pupil. So the iris is like the border between the anterior segment and the posterior segment. So the posterior segment back here has vitreous humor, which is actually so thick that, if you're careful, you can hold it in your hands. So yeah, there we go. So actually, you can't really see your neural layer in there. Why? Because it's microscopic. This model, I think, well, here it is. So, whoa! Not showing you the what? What's that? Optic disc. Oh, what do you imagine that is? Macula lutea. Okay, yeah, so inner layer. I guess that's the next part of the lab anyway. Let me scroll down here. Fovea centralis has only cones and is going to be where you have the most precise vision. Okay, here we go. Oh, this poor eyeball. So, here we go. If we are looking at this whole entire eyeball together, the lens sits here. Cornea, which is part of which segment? The anterior segment. Which layer? Fibrous layer. Sclera. And then she broke the eye. Okay. Cornea and sclera, fibrous layer. Okay. Vascular layer. It's right here and here. Okay. So that's the ciliary, like the whole ciliary apparatus. Ciliary body specifically is the muscles, and yeah, this model doesn't really, they should be lightened up in here. Has the suspensory ligaments that are going to grab onto your lens. This is a hot mess. Back here we have vitreous humor, which is right here. And this is why it's a hot mess. I need you to hold my lens. Just like it does in the body, right? The vitreous humor gives structure to the eye. It does so in your model that's falling apart. Two. It's supposed to be super glue. The super glue is not super. Anyway, vitreous humor, posterior segment, holds your retina to the back of your eye, your neural layer. So your retina has the rods and the cones. Look at this. I'm so glad that the professor brought this model home. So back here we can see our rods and cones that are talking to our bipolar cells, that are talking to our ganglion cells, and our ganglion cells are sending their axons back in what becomes the optic nerve. So how that works is that your rods and cones, so let's just imagine here that this is the back of the eye and that this model isn't a million years old. Your rods and your cones are back here, the most posterior aspect of the neural layer of the retina, right? Okay, and so then they're talking to these bipolar cells here, and the bipolar cells are talking to these ganglion cells, and the ganglion cells are shooting their axons back out the optic nerve. So the weird thing about that is that light comes in this direction and hits the retina and is processed in this direction, because it first hits the pigmented layer of the retina, which traps it, and then it is processed in the rods and cones that talk to the bipolar cells, that talk to the ganglion cells, who are shooting their axons back that way. It's pretty cool. Look at it in your book. So if we need a refresher, the light is coming in through your eye this way and hits it, and then it's processed this way so that my rods and cones tell my bipolar cells, tell my ganglion cells, and then the signal goes out. It's pretty cool. I was trying to figure out which model was best to see the extrinsic eye muscles on, and I think it's this one. So I know you've got so much to memorize, but one of the things that you memorize like first day of class or second day was like directional terms and stuff. Okay, so superior, what does that mean? Closer to the head, right? So here's your superior rectus and inferior rectus. Rectus is like straight, dudes. Okay, rectus muscles, their fibers are all running parallel to each other. Superior rectus, inferior rectus. Can you tell if this is a left or a right eyeball? You can't yet, but you'll be able to after I tell you what that thing is. It is that's not a Rona cough. It is the insertion for the superior oblique. Okay, so it's superior and it's at an angle. And here's your inferior that's at an angle. Oh, we don't need the whole eye to see the extrinsic muscles. That's the point. They're outside it. Okay, so this actually goes around, this muscle goes around and wraps around the trochlear nerve. What's the trochlear nerve do? It's motor to the eye, right? So, okay, so that's why we can't see this muscle. So if this is going around the trochlear nerve, that's inside your head. So this is actually a right eye. Alright, so that makes this superior rectus, inferior rectus, that makes this lateral rectus, and this medial rectus. That makes this inferior oblique. Inferior and at an angle, right? Because that's what oblique means. And this is our attachment for superior oblique, okay? Superior and inferior oblique. Angles. Look at that. Angles. Superior and inferior rectus. Look at that. Rectus. And medial and lateral rectus. And those, my friends, are your extrinsic eye muscles. Okay, so here's an ear. Auricle, helix, lobule. Funnel sound into the external acoustic meatus. When you're scratching your ear, you're digging out earwax. That's where it's happening. That is like not helpful at all. I didn't say I was helpful. Well, I am helpful, but not all the time. You have to take attitude and sass, because that's what you get from the assistant. Alright, so let's pull off some bones here. So inside your skull, your temporal bone is like got this whole tympanic part that's protecting your ear. It's awesome. Why did you do that? Like messing up my game here. Tympanic membrane. Okay, your sound. You are vibrating through space and being funneled into the external acoustic meatus. And you're hitting the tympanic membrane. And then what's happening is you are rocking the malleus, which is attached to the incus, which is attached to the stapes, and the stapes is, oh, awesome, and attached to this thing called the oval window. The oval window is knocking on this thing here, the vestibule. The vestibule leads into the semicircular canals, which are responsible for equilibrium and sensing head position, and the cochlea, which is responsible for hearing. Both of those things send action potentials through that bad boy, the vestibulocochlear nerve. Okay, so external ear leads into the middle ear. When the sound is coming through, it's going to hit the tympanic membrane, which is right here. I like the other model's tympanic membrane better because this is more what it's like in the body. A clear window. And that means your lecture's done processing, but I have nothing to do with that. I'm the assistant. So this clear window is going to knock on the malleus, which is going to rock the incus, which is going to knock on the stapes, which is broken on this model, which is why we're looking at this model. So the stapes is this super cool bone right there that's knocking on the oval window. And you can never see the oval window because it's deep to the stapes. Okay, so the other thing that you have to know here is the Faringo tympanic tube. And if you've ever heard, like, or if you know, when you pop your ears, when you get all that pressure and you pop your ears, all that pressure is being let out through your Faringo tympanic tube. It's also why you can get like a sinus infection in your nose that then like goes into your throat and then somehow winds up in your ears and it's a hot mess. It's because it's all connected. And that's the big like bumper sticker of life. It's all connected. Okay, so that's out of the middle ear, and now when we get to the inner ear, you guys are only focusing on hearing really in class, and that is all occurring here in the cochlea. So the cool thing about this is if you were to like unwind it, you would see that you've got hairs of different lengths that are responding to sound waves of different pitches or vibrations. Vibrate different parts of the what membrane? Basilar membrane. And so, yeah, that will rock those hair cells and agitate them, and they'll generate and action potential along this cochlear branch of the vestibular cochlear nerve. Okay, so, vestibule is like right here. Oh, entrances. Vestibules in churches are entrances. Vestibule to your ear is the entrance. And it leads into the semicircular canals that are responsible for our head position and equilibrium. Dang it all right to heck. I was hoping I had the other model's semicircular canals here, because in the other model you can see the bony and the membranous labyrinth. Right now all you see is the bony labyrinth. And it's this bony construct that looks just like this that's protecting all of this fluid-filled membrane stuff that is vibrating in response to different pitches. Also, if there are sound waves that we don't respond to, things that we can't hear, like things that dogs could hear but we can't hear, those sound waves don't rock the basilar membrane and the pressure is let out through here. So things we can hear rock this and it goes back along here, but then in here you have these sensors that as you tip your head, so like just imagine this is your head, and as you tip it every which way, there are these receptors in those semicircular canals that are sending signals back along the vestibular branch to let you know about your head position. So those are the semicircular canals. That's the vestibule, and that's the vestibular cochlear nerve, and it exits through the internal acoustic meatus, which you'll be looking at when you look at the skull. So that's the end of this lab. See you next time, tomorrow, for, which way is peace for you guys? This way. tomorrow for some eyeball dissection from the kitchen. Yeah.